Hello, everybody. My name is George Huang. I'll be talking about a study that I've worked in collaboration with the students and colleagues in Taiwan. That was where I came from. And this is really a study looking at environmental source of the human bacterial pathogen C. difficile in water samples. So I have another poster talk where I introduce about C. difficile already. So I just go really quickly. So C. difficile is a bacterial pathogen. It forms spores are highly stress resistant. And C. difficile can cause disease in people whose gut microbiota has been compromised, either because they are immunodeficient and also because they are using broad spectrum antibiotics. I'll talk more about that later. The symptoms of C. difficile infections can range from mild diarrhea to more toxic megacolons to sepsis. However, one of the most difficult problems with C. difficile treatment is that because of the spores are very highly resistant, and C. difficile are also known to be resistant to many antibiotics, a lot of patients have multiple relapses. Here is an overview of the transmission cycle of C. difficile. So basically, C. difficile can reside in animals, in humans, in soil as well. And when we ingest C. difficile spores, they usually reside in our gut. And they don't usually do anything because their growth is inhibited by the gut commensal. However, if we use antibiotics, these antibiotics will kill off all these regular gut commensals, and C. difficile being resistant to them will then quickly outgrow, produce toxins, and cause these symptoms that we see associated with the infection. And in addition, they'll undergo sporulation again as well, and these spores then can be released through the fecal materials of these hosts and into the environment again, and thus the cycle continues. Okay, now so for this project, we were interested in looking at what about water source samples. There has been some reports that C. difficile can survive in the water, but we don't really know whether this is actually a very important source of C. difficile that we'll find in human patients. So for this study, the students went and looked at the wastewater from hospitals as well as from city wastewater treatment plants. And the hypothesis is that spores, because it's so stress resistant, it can probably survive whatever they do in these water treatment plants and also from the hospital. In addition, we hypothesize perhaps these C. difficile from the wastewater might go and contaminate farmed seafood such as oyster and clams. Okay, so here the students collected waters from these samples and as well as seafood, concentrated the samples using a pump and filter membrane, and then through alcohol shock and heat shock, we can get rid of non-sporulating bacteria, and then we use a selective medium called CCFA to isolate C. difficile, and then PCR is performed to confirm they are C. diff, and then we perform our downstream analysis. Okay, and right here is a summary of the results. A total of 97 different isolates of C. difficile was obtained in this study. And these are the spread depending on the source, whether it's the hospital, the wastewater treatment plant, and the seafood. The majority of samples actually we found are actually from the hospital, which isn't that surprising given that their wastewater treatment probably isn't as thorough as the ones in the city water. Okay. Now, here is an example of the results of the different genotyping analysis. So first, I need to talk about something called ribotyping. So the way we can differentiate different C. difficile isolates is through different length and spaces in the intergenic region of its ribosomal DNA region. Ribosomal RNA region, okay? And so we can, through a multiplex PCR, to amplify these regions, run it on a gel, and then the DNA fingerprinting pattern can then assign a particular isolate to a different type of ribotype. Okay, and so just to make this really long story short, we found interestingly that there's two different ribotypes that we commonly see in human. We identify in the water source as well. One's called ribotype 027, which is a known global pandemic strain already. The second one belongs to a sublineage of what's called ribotype 07A. Now 07A is what we call the hypervariant strain because it's known to be more resistant to antibiotics as well as produce more toxins. So people who are infected with these strains tend to exhibit more severe disease. Okay, so here's an example of an experiment where we're looking at the antimicrobial resistant profile of these strains. Again, I've highlighted in red these ribotype 027 and the 07A lineages of C. difficile. You can see they exhibit basically resistance to fluoroquinolones, and that's indicated right here: moxifloxacin, levofloxacin, ciprofloxacin. Okay, and so this is one of the reasons they are more prevalent in humans, because they are resistant to these broadly used antibiotics, in addition to the toxins that they produce as well. And again, we were really surprised to find basically all the isolates we found that come out of the hospital belongs to these more hypervariant strains, and that was quite surprising as well. Okay, and then we also look at other phenotypes. One of the phenotypes bacteria have is so-called Flagella swimming motility. And to make a long story short, we were able to find for the first time, this never been reported before, that C. difficile that belong to the 07A lineage families of C. difficile actually have less ability to exhibit swimming motility. And we don't know why. We do know in the literature that there are regulators, transcriptional regulators, that regulate both toxin production as well as Flagella motility. But exactly how that tie into the pathogenesis is still unknown and under investigation. Okay, and lastly, this is our proposed transmission cycle. We believe that the C. difficile that are shed either through symptomatic or even asymptomatic carriers, these spores are not going to be treated, are not going to be killed off by the wastewater treatment plants. Okay, and then because of this, these spores can actually be released into the waters that are floating around. And in some cases, they could probably also contaminate the downstream farm animals. And whether the seafood are actually a potential source of C. difficile infection, it still remains to be investigated. And that's all we have for here. Thank you, and I welcome any questions.